Welcome, madam. So we had started, we were waiting for you, madam. So again, good afternoon, you all. So we will start today's technical session now. I think I am audible. And to start with, we are really delighted to have with us Dr. Aparna Mukherjee. Dr. Aparna Mukherjee is scientist F, clinician, scientist fellow, in charge of clinical studies and trials unit at the Division of Developmental Research, ICMR, New Delhi, Department of Health Research of Ministry of Health and Family Welfare. We are really delighted to have Madam with us. I request you all to post your questions on the chat box, and we will take those questions at the end of the Madam's presentation. Madam will tell us about the clinical trials in infectious disease, public health priorities. Welcome, Madam. Now this stage is yours.
Thank you for your kind words and thank you for inviting me. I'm sure you have a very interesting and learned audience who we are being able to reach through this. So I'll share my screen. Can you see my screen?
We can see your screen, madam, but the presentation I cannot see.
Okay, I'll just stop sharing.
Yeah. No, it was coming. Your presentation.
Okay, so maybe the net is a bit slow. I'll just get it. Now you can see it?
Yes, it is full screen also, madam.
Right.
Thank you.
Okay. So it's more of, I mean, I have a few slides, but I thought that this would be more like a, discussion regarding clinical trials in infectious diseases. And because the trial environment right now, the ecosystem in India is something which is really blooming and really being encouraged. So we did have a slump in clinical trials, which we witnessed around 2012, 2014, when people kind of, averse to doing trials, as well as participants averse to getting enrolled in trial because there was a lot of misgivings regarding trials. But as of the current ecosystem, as we can see, is that there is a renewed interest in trials, renewed interest in both conducting, participating, and I would also say funding trials. Because clinical trials, as we know, is the most important cornerstone. It's the best way to gather evidence, whether not only in infectious disease, in any disease that you think of. So what is the scope of these studies that we have in infectious disease? We could have diagnostic studies, trials of a lot of diagnostic algorithms, diagnostic technologies, and we also know medtech is something that is being hugely encouraged in the research ecosystem right now. So infectious disease diagnosis is again one of the main pillars of these medtech innovations. So they need to be validated, they need to be studied. Then we have a whole plethora of preventive, preventive strategies, be it again, bundles, sepsis bundles or interventions which are behavioral, or we talk of vaccines. So in infectious disease, we have a full spectrum of preventive therapy that we can work on. And of course, we have the therapeutics, which could be small molecules, which could be biologics, which could be our, immunoglobulins, etc. So we again have to have a whole spectrum of therapeutics where we are looking into the safety and efficacy of these therapies, if it's a innovative drug, or we are looking into repurposing it for a new indication. And at the same time, we have all the phases that we can go through. Phase one, where you are primarily looking into the safety, looking into the first in human studies, and up to the second, third, the more definitive trials that do. And the current buzzword in the pharma industry is whether we can actually interpret something from real world evidence. Because as we know, there are number of patients that we treat with many of these, when we're talking about off-label treatment or we are talking of repurposing drugs, we do have a plethora of information with us as clinicians, as treating physicians. So there is a renewed interest as to whether we can use this real-world evidence in a systematic way, in a statistically understandable way to actually come to some conclusion. So these are the various ways that you could do clinical trials in infectious disease. And as you all are aware, COVID-19 has been a watershed moment in research around, be it India or globally, particularly infectious disease. Like if you just see, this is a paper that was published in 2019, that was just before before the pandemic struck, and they did a review of all infectious disease related trials that were registered in clinicaltrials.gov in the last decade from 7 to 17. And they found that ID-related trials only represented 6.9 to 9.9% of the trials that were registered, and that was kind of plateauing over the decade. It's not like it was increasing. But then COVID hit, and we saw an exponential rise in trials related to infectious disease. I don't think so much of research had ever been done that was done in those 2 years. But that did leave us with a lot of learnings and a lot of, lot of warnings as to how we should not do research. And at the same time, a lot of lessons and learnings as to how we can actually better our research. So if I can take you through what are the challenges of clinical trials in ID? Like I just thought that clinical trials is something that you know of, you have already heard of, you might be doing, you might be conducting, reviewing, being part of. But so what is so special about clinical trials in infectious disease? Because infectious disease, as we know, is a major chunk of the patients that we see in India still today. Even if NCDs are rising, they are causing major, large chunk of the mortality and morbidity is the main reason of mortality in many of the states, but still infectious disease is there in our daily practice. So what is special about doing studies? But as we saw in that decade-long systematic trial, a systematic review, trials, though it's a high burden condition in LMICs, the proportion of studies, or I shouldn't say studies, the proportion of actually clinical trials that are done on infectious disease are limited. You'll get more trials in the non-communicable disease sections, diabetes, hypertension, etc., than you will find in, ARI or diarrheal diseases. Though you will find a chunk in high-profile diseases like HIV, hepatitis, etc. So what are the challenges? One is that it's a dynamic field. You have emerging, re-emerging infections, right? Though in our country, we cannot call TB, dengue, malaria as either emerging or re-emerging. They've always been there, and now we are fighting to kind of eliminate them. But we do have a lot of other diseases like Ebola, like COVID-19 that we had, other vector-borne diseases. So there is always a cycle where you get newer diseases, less known diseases, which we have to work on. And then definitely we have these outbreak situations, pandemic situations, which in itself has a lot of roadblocks and difficulties in doing trials. And the other way is that if you're talking about a pathogen which causes an outbreak, I mean, say Ebola, you have a vaccine for that. Now, where do you test it, right? So you can test it. You can go to the endpoints of immunogenicity, reactogenicity, but you will never be able to go to the clinical endpoint of whether it is preventing the disease or not. Say we have a vaccine come. We can go to the endpoint of having, the neutralizing antibodies, the immunogenicity, but we will not be able to go to the clinical endpoint to say that whether it's actually preventing the disease or not, isn't it? That is a very big challenge in these kinds of diseases, which occur only in outbreaks and are not endemic, will not be found in general population. Then again, as I said, that there is a high burden of infectious disease in LMICs, but then we are very fortunate now that we do have government funding. We have a lot of other avenues of funding in India. But if you talk of the global funding, the funding that comes from the North, the global health funding, as we say, they don't have the burden of infectious disease, right? But the burden is in our countries. So the ideas have to come from us in order to do trials in infectious disease rather than from the global North. So even if you talk of the pharma industry, which is a major driver of such clinical trials, they have less interest in, say, newer antibiotics. I mean, a new antibiotic, there's been ages since we are having newer antibiotics in spite of AMR that we are seeing. In cases of neglected tropical disease, pharma industry has got very, very less interest in this. It's the philanthropic organizations, research organizations, which mainly drive the research here. So there is a lack of funding opportunities, but again, this is changing in time. So what other challenges would you face in clinical trials when you do it on infectious disease? Of course, you are dealing with a vulnerable population, even if it's not an outbreak, if it's an endemic condition. Usually we would find that the extremes of ages are affected badly. Immunocompromised people are affected badly by these. So all these groups in themselves are vulnerable population, and you have to take extra precautions, and you have to be more careful when you are dealing with this group in terms of taking consent, in terms of taking assent, in terms of dealing with their complications, with their adverse events. So, so in general, you will be dealing with vulnerable populations more than in other streams. And of course, since you're dealing with infectious material, you are dealing with patients who can potentially infect you. You have to follow all the universal precautions as well as pathogen-specific precautions, like we saw during the COVID-19. Research was difficult because of limited access to the patients. You had to have all your protective gear, and only then you were allowed. And even then, the access was very, very limited at least during the initial phase. And then there is always a changing pattern of the pathogens. That is true for all diseases. Even diabetes has kind of, you would say, evolved over generations. But then this kind of a mutating strains that you find in case of HIV, the antigenic shift that you find in influenza, it's very, very characteristic of infectious disease, where actually testing vaccines, testing newer therapies are very challenging because of this mutating habits. Another very interesting point that you should know about infectious disease is the endpoints, because when you design a clinical trial, of course, asking a good research question is the cornerstone of having a good study, right? And one part of the research question is, how do you measure the outcome? What is your outcome measure? What is your endpoint? So usually in infectious diseases, when we are, testing a vaccine or testing a new drug or even a repurposed drug, many a times we depend on immunological, in these surrogate endpoints, of protection or of efficacy. It may be immunological endpoints, whether the antibody titers are rising, or microbiological endpoint, whether cultures are becoming negative. You have early bactericidal action in case of TB drugs. But we have seen that in many cases, even if these endpoints are reached, it may not always translate into actual cure of the disease or a improve clinical endpoint. So what would be more beneficial or of interest to both the physician and the patient would be a clinical endpoint, an endpoint which actually shows an improvement in the patient. It could be a reduction in mortality, it could be reduction in hospitalization, reduction in the symptoms. But those endpoints are usually difficult to achieve. They take longer time. You're doing a TB disease. If you want to actually see clearance of X-ray, clearance of all symptoms, you have and prevention of relapse, you may have to give a full 24 months to see that. So in that, usually what we do is we take these surrogate points. But for your research to be meaningful, it is important that you keep in mind that the clinical endpoints, the hard endpoints, which actually will translate to improved practice and policies, are always preferable, and they even if your study takes more time, is more difficult to do, it's always advisable to do that, because that will be a more decisive study than rather if you just take a biochemical or immunological marker to say that, well, this has worked or this has not worked. Then again, post-trial access, that this is not unique for infectious disease. Post-trial access is something that we keep discussing even in other conditions, like in cancer oncotherapy trials, where we would like to say that if the drug is effective, people who did not get it, maybe were in the control arm, should receive it after the study has been over, or they should get continued treatment after the trial. Same thing is applicable for infectious disease also, particularly in vaccine trials. We need to have a provision that if the vaccine is found to be effective, the control arm, which did not get the vaccine, should actually be vaccinated after the study is over, and you know that this particular modality is effective, is efficient, and is safe. So that is something that should be inbuilt in our, in our agreements and in the way we design the trial. And we, particularly when we are doing sponsored trials, industry-sponsored trials, we need to negotiate with them to say that, yes, post-trial access has to be provided. Community engagement, again, as I said, is not unique for infectious disease. But since a lot of these studies would be done in the community, you're doing a vaccine trial, you need to recruit your healthy volunteers from the community. So you need to have engagement. And that engagement only does not mean that you kind of give them incentives or anything like that. The engagement should start from before you design the trial, that whether this particular trial, many a time scientific questions are asked because we want to ask them as researchers, as clinicians. But we also need, the mindset is changing, and now we also realize that we need to ask the community, we need to ask the participants whether it will be helpful, whether you want, what kind of questions do you want answered, what kind of endpoints would be really beneficial for you. So that amount of community engagement, it's not that common in our country as of now, or I would say it's not common globally as of now, but it's something that we should endeavor towards, we should work towards. And we do have these community advisory boards now, particularly in cases of sensitive, conditions like HIV, or if you are dealing even with TB patients, chronic diseases, forming a community advisory board where you have patient advocates, you have representations from the community is advisable, because they will help you tell how you can improve your recruitment, your retention, because keeping participants in trials, particularly if you have a long follow-up, is a skill set that you need to develop as a trialist, and good community engagement always helps in that. So, I mean, these were some of the challenges that came to my mind and for infectious disease, and I'm sure all of you who work in the infectious disease domain will have faced many other challenges, and you're welcome to add in the chat if any others that you think of. Then we can always come to the pandemic, which I said has been a watershed moment in all our lives, be it as general population, be it as clinicians, or be it as researchers. So there were very pertinent problems that we faced during the pandemic doing research. There was a lot of research done, a lot of resources that were used. And there were good findings. We had vaccines, we had drugs, but there was a lot of research waste also, in the sense that there were a lot of smaller, disjointed studies done, which may not have proved decisive, and many of them did not even complete. Like all the convalescent plasma trials that were started in America and Europe could not, many of them could not finish their enrollment, could not complete their enrollment even before a decisive, treatment strategy had arrived. So what were the challenges that we faced? It was a brand new pathogen. We did not know how it worked or what worked against it. So we were learning as we went. So that, of course, that was the greatest obstacle. And at the same time, we needed rapid response. It was spreading rapidly, killing rapidly. So of course, we needed a rapid response. Usually we take a time to design a trial, do a trial, though we would always say a successful trial has two things in it going in its favor. One is timeliness. It should have a timely completion and the quality of the trial. But here, both of these things were like kind of tested and challenged to the extreme. We had to be very fast, and at the same time, we had to maintain quality in very difficult circumstances. A lot of repurposed drugs were being tested because we didn't know which would work, and that led to actually develop, use of a lot of newer ways of doing trials, like we had adaptive trials, platform trials, which were not so commonly used before COVID-19. And of course, the logistics were a nightmare. There was lockdown, there was difficulty in access to patients because the patients were kept separate from the rest of the hospitals. There was difficulty in transporting samples or medicines or kits. So all these things had to be overcome during that period. And at the same time, there was a hesitancy in participation also, because even as general population, we knew that even the doctors don't know what is working or not. So participating in a trial was something that everybody hesitated in, and particularly during follow-up. Once people were discharged from the hospitals, they did not want to come back to a healthcare provider or to a hospital for any kind of follow-up. So follow-up studies with the COVID-19 positive patients were very, very difficult. So these were some of the challenges that we faced during pandemic. But at the same time, we did learn a lot from this pandemic. As I said, a lot of the way that we do research, that we view research now has changed, has evolved because of the pandemic. Collaboration can work so well, national as well as international, that we saw during the COVID time. We had the public, private, all kinds of enterprises coming together to make the vaccine, to make the other therapeutics. And hopefully many of these collaborations have survived the test of time, and they are continuing even post-COVID. Also, we realize the importance of ready networks that can actually conduct clinical trials. Choosing sites, getting them ready, and then starting a trial takes time. So the countries which actually had these existing networks, which they could pivot, like UK, they could pivot their existing network of NIHR institutes into doing the large recovery trial. In days, actually, they could start. So that was very important. And all funding organizations realized the importance of ready network. Then also we got to learn ourselves and got to use these newer techniques like decentralized or virtual monitoring, right? Till now, we would always say that it's a personal monitoring, in-person visits are always better than anything virtual. But COVID taught us to do everything on Zoom, including like we are doing this webinar now. I'm sure before COVID, this would have been an in-person webinar. So we did all our trials, we monitored them, starting from site feasibility, video calls were done to see the site feasibility, audits and regular monitoring, everything was done virtually. So it was like not by choice, but because we had to do it. So a type of decentralized trial system was developed by most of the researchers. And as I said, newer designs like adaptive trials, platform trials like the recovery, they all came into fruition. These designs were there. It's not that the designs were new. They have been there for ages, but they are mainly used in the early phases of drug discovery or in oncotherapies. They were not so commonly used like now we are talking about them so frequently. In fact, our regulators, our ethics committees, they were also not actually equipped to, review a master protocol and see that there would be multiple treatments, multiple randomizations. The whole thing took some learning on the part of both the reviewers, the regulators, as well as the researchers to get it going. But we can say that post-COVID, we all are more experienced trialists. We have these newer collaborations, newer tools in our hand to be able to do more effective and robust trials. So since we're talking of priority health topics, so infectious disease prioritization is done by many of the funders because we all know that resources are limited. There's only so much resource that can be spent on research. And so we need to know what are the pathogens that we need to focus on, right? So most of the research organizations, funding organizations do their own prioritization exercises. Like WHO, their R&D blueprints, they update this to recognize the pathogens which have epidemic potential. At the same time, where they don't have enough countermeasures so that more research is needed. So this is the present list that they have. COVID-19 is still there. Then we have Crimean Congo hemorrhagic fever, Ebola, Lassa, MERS and SARS. Nipah virus is there. So, we are trying to work on Nipah viruses. Rift Valley fever, Zika. We're also trying to work on Zika virus. And disease X is obviously the probable disease that might cause the next pandemic. So ICMR also has its own list of national health research priorities, which has been developed with a prioritization exercise and which matches with the national health priorities of the government of India. And there are about 11 to now we have increased it to 13 priorities, out of which infectious disease related ones pertain to antimicrobial resistance, One Health, tuberculosis, and all the vector-borne diseases. So obviously the focus are on these infectious diseases. Wellcome Trust, which we know is a important funder globally, also they have their prioritization list to focus on drug-resistant infections, including tuberculosis and fungal diseases which are impacted by environmental changes like the arboviruses, schistosomiasis, cholera, etc., and of course, AMR. So these prioritization lists, as researchers, they help us actually to see where the focus of the global community or the national community is, and to align our research priorities, if possible, to these health priorities. Lastly, I would say that the clinical trial ecosystem in India right now is quite conducive for trial conduct. So it's a very good time to be a trialist in India, actually, because there is encouragement from funding agencies, from government to actually conduct trials, because we have realized that we are a huge population. We have diversity in population. We have the load of the disease, and we have the need to find our own answers. But A very small proportion, about 8 to 9% only of the global clinical trials are actually done in India. And there is no reason why we can't change that. Not only because it's like we need to be a part of the global research system, research community, but then that way we will be able to find answers for the solution, for the questions that we can ask for our own problems, rather than depending upon the global north to ask the questions and give us the answers. So right now we have these enabling, rules and guidelines. You all must be aware of the new Drugs and Clinical Trial Rules 2019, which is a very comprehensive document telling us how to do the phase 1, 2, 3 trials, what are the submissions required. And we now have a very facilitative regulatory agency also, in the sense that they are trying to kind of shorten their approval times. They have a fully electronic submission, which takes away all the need for up and down, coming to Delhi or submitting, etc., etc. They have regular SCCs. They are trying to increase their pool of experts so that the newer therapies like stem and cell and gene therapies or newer biologics can also be evaluated properly. And for example, they have ICMR has deputed scientists from our cadre to the CDSU so that to give them a boost for their SECs also. Then we have a very good document, which is from ICMR, which is the Ethical Guidelines for Biomedical Research and Human Participants, which tells you a lot about how you can conduct studies ethically. Because after all, we have to remember that we are dealing with human participants. So however interesting a question may be, we have to weigh it to the risk-benefit ratio, and the benefit should always be more for the participants and the risk. So this is a very important document that you need to follow if you're a trialist. GCP, of course, the ICH has just undergone a revision. We have the R3, which is taken into newer aspects like risk-based monitoring and more of decentralization, etc. There is an Indian GCP, which is published by CDSCO, which is quite kind of dated, but then it has been recently updated. I'm sure the updated version will be published very soon. And as I said, there is a renewed focus on trials in terms of the funding agencies, in terms of the interest in the government. So grants like, as you must be aware, even ICMR grants, now there is a huge focus on finding solutions rather than only describing the problems. So trials get a lot of grants. DBT also has their own networks and provisions of giving grants, particularly to innovative drugs. Then coming to networks, as I said, COVID-19 showed us that we needed networks. We needed ready sites which can work as a plug and play solutions. Whenever we have the need, they can start a multi-center trials. So ICMR has developed a network where there are 4 phase 1 centers, which is a very unique endeavor from the part of the from our part, so that we are having phase 1 centers in the public sector. Then we also have a 75 site strong intent network, which is primarily for the late phase trials. And besides that, as I said, DBT also has their own networks, and disease-specific networks are always available to you. Then we have provisions for CTRI registration, which is the Clinical Trial Registry of India, which is WHO compliant, and it is essential for your publications, for getting grants, that all your trials are prospectively registered. That is, the registration is done before the first patient is enrolled. If you try to register retrospectively after your study has started, that is not possible at this time and date. And I would also request researchers who actually register in CTRI to complete their registration, in the sense that when the study is over, you should upload your reports, you should upload the status of your recruitment, because this is basically a archival. It's basically a data mine for everybody to see what trials are going on in a particular field. And if we have the complete result, that will tell us as to whether the trial was successful, whether it should be repeated, whether we need something else. So in order to have that transparency, it is very important that we kind of upload the results also. So this was in a nutshell that we had regarding the infectious disease ecosystem in our country. Thank you. And if there are any questions, I'll be happy to take them.
Thank you, madam. Thank you for your great deliberation. I think two important take-home message, what I gathered, and I want to tell all the respected delegates who are attending. Many of you are postgraduate students of medicine or pharmacology. Madam had emphasized on the role of decentralized trials. So it is very interesting and important options for us to become clinical trialist. So all of you, please try to learn about clinical trials. It's A to Z. And I think I have one specific question to you, madam. Before that, I want to extend my heartfelt congratulations to you for your leadership in the landmark Placid trial, and it was being published in BMJ. I think India's first multi-center randomized control trial on convalescent plasma during the COVID-19 pandemic. I think three important points what we get from Placid trial set that set an important benchmarks. Number one, it was a pragmatic real-world clinical trial design across public and private hospitals. Number two, rapid implementation under emergency health conditions. And number three, evidence-based reassessment of a widely used therapy during a global crisis. So, madam, my question is, what were the core design elements? I think this could be a very bigger answer, but you can give us one summary. That what were the core design elements of this Placid trial that made it feasible across almost 39 or 40 diverse clinical settings in India? And how can this be replicated in future pragmatic trials?
Right. Thank you for that. Actually, Placid was the first trial globally which was completed on convalescent plasma. As I said, during that time, it was very difficult to enroll patients. So many of the trials that started could not complete the enrollment. They had to kind of abandon the trials in between. So this was the first one which was completed within time. So one, definitely it was because of the emergency, because of the outbreak, all the medical colleges, investigators, they kind of showed interest, they came together. But as I said, this kind of a network is very important. If you have the existing collaboration where you know that these centers are capable of doing a phase 3 trial, then it is much easier to start a trial rather than if you have to start from scratch and identify centers across the country which have the capacity, which have the manpower and the logistics to do a trial. So that is one of the most important takeaway that we had from the trial, COVID trial, and the other, not only Placid, all the other COVID trials, and because of which we really started our phase 1 as well as phase 2, 3 networks. And definitely, as I said, it's a very simple randomized controlled trial, which kind of looked into convalescent plasma versus standard of care. But the essence was to finish it in time so that we have some answers, while it is still important to have the answers. And the essence was to actually coordinate all the 39 centers. So that's where our decentralization or virtual monitoring and that kind of coordination helped a lot.
Yeah, thank you. Thank you, madam. One question I just want to ask, because that has been in our chat box. I think as we were discussing about clinical trials in infectious disease, specifically, what is your take on CHIM model? CHIM, we all know that controlled human infection model studies, CHIM studies. So what are the specific or scientific justifications for using CHIM models in accelerating, especially in vaccine development for different endemic disease endemic infectious disease in India? What is the current situation, madam, here?
Current situation is we do have a guideline, ethics guideline as to how to conduct these, but it's not that we are actually implementing it regularly, right? Because it is a very difficult model to follow in the sense that actually causing the disease, causing it in, causing the disease in healthy volunteers, and then trying to see there are always two sides, two schools of opinion, which would always argue whether this is at all needed or not. But yes, you have to do it in a setting where you are ready to you have countermeasures for whatever is the fallout of that disease induction, and if your therapy doesn't work, if your vaccine doesn't work, then you should be able to, counteract that measure. So that provision should be there. Otherwise, embarking on this is very difficult. As of now, India, it's a theoretical proposition. We really don't have trials that are using this model.
Thank you, madam. I think we don't have any more questions. Let me just check once again the yeah. So there is no further question as of now. I request all the delegates, if you have any question, you can type that and we can ask madam via email, and we also can get back to you all if the madam when the madam will give us the
I'll be
answer.
happy to answer.
Yeah. So once again, thank you very much, madam, for giving us your valuable time and for this enlightening lecture. I think many of us had a huge benefit upon your lecture. So thank you, Professor Mukherjee,